My name is King Jordan. I'm a faculty member in the School of Biological Sciences at the Georgia Institute of Technology in Atlanta, Georgia. And I'm going to talk about how genomic ancestry inference can be used to decompose genetic and environmental contributions to cancer disparities. So the U.S. National Cancer Institute defines cancer health disparities as higher rates of new diagnoses and cancer death rates among certain races, ethnicities, or other population groups. And unfortunately, cancer health disparities remain both a pressing social and scientific problem. Even though the overall rates of cancer incidence and mortality are declining across all population groups in the United States, for certain groups, there continues to be an increased risk of developing or dying from particular cancers. And examples of this include the fact that Black men and women have higher death rates than all other racial or ethnic groups for many, although not all cancer types. In addition, despite having similar rates of breast cancer, Black women are more likely than white women to die of the disease. Black men are twice as likely as white men to die of prostate cancer and continue to have the highest prostate cancer mortality among all U.S. population groups. Hispanic and Black women have higher rates of cervical cancer than women of other racial or ethnic groups, with Black women having the highest rates of death from the disease. American Indians, Alaska Natives have higher death rates from kidney cancer than any other racial ethnic group. And American Indians, Alaska Natives also have the highest rates of liver and intrahepatic bile duct cancer, followed by Hispanics and Asians, Pacific Islanders. So these are just some examples of cancer disparities that persist in the US population. Now, the causes of cancer disparities are complex and multifactorial, with varying contributions from genetic, social, and environmental risk factors. So there can be genetic and biological factors that influence the observed disparities, as well as differences between groups with respect to access to healthcare, socioeconomic factors, environmental exposures, diet, and lifestyle. Again, all of these factors can contribute to the differences that we see in the incidence and death rates of cancer among population groups in the United States. And what we believe, what I'm posing here today, is that genomic ancestry inference can be used to help decompose the genetic, social, and environmental contributions to cancer health disparities in the US and other cosmopolitan diverse countries. So when I say ancestry, what I'm referring to are the geographic origins of a person's ancestors. So in a cosmopolitan diverse country like the United States, people can have ancestors from many different parts of the world, and where our ancestors are from impacts our health and health disparities, like cancer disparities, in a variety of different ways. And we can think about ancestry as being defined in two related but really distinct ways. And one is socially defined ancestry, and that is the race and ethnic groups that I referred to when I defined cancer disparities. But then another way to define ancestry is via genetic inference. So genetically inferred ancestry is a characteristic of the genome. The story of our ancestors, where they're from, is written in our DNA. Both of these dimensions of ancestry are important for health and relevant to health disparities. So socially defined ancestry impacts individuals' health with respect to differences in the social environment and lived experiences, including but not limited to structural inequities that we see between different groups, environmental injustice, and differences in access to healthcare among groups. Genetically defined ancestry impacts health largely via differences in the frequencies of health-related genetic variants. So these are genetic variants that are associated with the risk of disease, but also genetic variants that influence how individuals respond to medication. So one of the big challenges for health disparities research in general and for cancer disparities research is the fact that the social and genetic dimensions of health co-vary across racial and ethnic groups. And thus it's difficult to tease apart their effects if we want to understand what the causes of these disparities are. And that's where genetically inferred ancestry comes in. So as I mentioned before, genetically inferred ancestry is a characteristic of the genome. It's written in our DNA. And what this means is that it can be inferred independently of the social dimensions of race and ethnicity. Unlike the social categories of race and ethnicity, we can infer genetic ancestry objectively and with precision. We can infer it both as a categorical or a continuous variable at different levels of relatedness. So at the continental level or at a finer scale of resolution, looking at the origins of individuals' ancestors within continental groups. And we can look at it at different levels of resolution. We can characterize ancestry at the level of the whole genome, genome-wide ancestry, or for specific locations along the chromosome, we can characterize their ancestral origins. So as an example of how we've been using genetic ancestry to look at the causes of health disparities is a paper that we published in Cancer Research last year entitled Association of Genetic Ancestry and Molecular Signatures with Cancer Survival Disparities. And this work entailed an analysis of the National Cancer Institute's Cancer Genome Atlas. So this is a large database containing patient and participant information for more than 30 different cancer types. So this was very much a pan-cancer analysis rather than an analysis focused on a single cancer type. And what we did for this study is we stratified individuals with respect to their self-identified race and ethnicity: Black, Hispanic, Asian, and white, the four largest race and ethnic groups in the TCGA, the Cancer Genome Atlas. And we characterized individuals' genetic ancestry based on their DNA variation. Then we looked at the relationship between ancestry and cancer survivorship, doing survivorship analysis. And then after that, we looked at a number of different molecular signatures. So we looked at rates of somatic mutation in cancer. We looked at differences in gene expression between normal and cancer tissue. And we looked at differences in epigenetic modification, specifically DNA methylation. Having defined the differences in survivorship between groups and molecular signatures, we then looked for interactions whereby genes' molecular signatures interacted with genetic ancestry to influence survivorship. In other words, we looked at cases where there were differences in survivorship between ancestry groups that were mediated by different genes' molecular signatures. And we found a number of interesting cases, including genes that were enriched in pathways that are related to the epithelial to mesenchymal transition, which is very important in cancer metastasis. This included genes and pathways that were related to recruitment of immune cells, including inflammatory cells, and related to inflammation via the action of pro-inflammatory mediators, cytokines and chemokines, as well as genes and pathways that were related to angiogenesis. All of these are very important in the progression of cancer and in particular metastasis of cancer. And ancestry influences genes vis-a-vis those molecular signatures that I mentioned in a number of different ways in the case of this study. And so what we found in this multi-cancer, this pan-cancer analysis, was 4 main cancer types with cancer survival disparities and 7 cancer-related genes that interact with genetic ancestry and thereby contribute to the observed survival disparities. Interestingly, we found that the regulatory changes that we observed that are related to differences in genetic ancestry and survival are mediated by epigenetic mechanisms rather than DNA mutation per se. So epigenetic mechanisms, DNA methylation in particular, have a greater contribution to cancer survival disparities than population-specific mutations. So overall, we uncovered various molecular mechanisms through which genetic ancestry impacts cancer survival disparities, thereby revealing potential population-specific therapeutic targets for groups that are disproportionately burdened by different kinds of cancer. And so just to close out what I perceive to be some of the challenges and opportunities for cancer health disparities research, first of all, we really need more data from diverse underrepresented populations. At this time, population biobanks and cancer registries still have a real bias for patients with European ancestry. We need better methods so that insights from current studies can be applied across populations or ancestries. And last but not least, we really need to develop human capacity so that investigators from diverse backgrounds can contribute to cancer disparities research. And with that, I will just close out by thanking the researchers from Georgia Tech, the National Institute on Minority Health and Health Disparities, and the Applied Bioinformatics Laboratory that contributed to this research, along with the entities that made the work possible. Thank you.